I am not sure if Prof is going to join, but I think we'll just proceed. I think we already have quorum. So by the end of this talk, we need to be able to list some of the oncogenic viruses, the known ones, to understand how RNA viruses mediate oncogenesis, and also understand how DNA viruses mediate oncogenesis. So viruses are etiological factors in the development of so many cancers. For example, cervical cancer and liver cancer. And historically, this guy by the name Peyton Rouse was able to demonstrate this, whereby he was able to inject foul sarcoma infiltrates into birds, and he was able to demonstrate that the RNA virus could cause tumors. So at least about 15 to 20% of all human tumors worldwide, they have a viral cause, but there's no single mechanism by which these viruses cause tumors. There are a couple of factors that are usually involved in the formation of the tumor. So it can't be the virus can't be the only factor. So generally, carcinogenesis involves multiple processes. As I have said before, the virus might be the initiating factor, but it is not the only factor. And also the processes, it might not be just a single process, might be so many processes involved. And as we've said before, the tumor viruses are not also complete carcinogens on their own. They act as initiators of the process, and different viruses that cause cancers have different mechanisms for immortalizing the cells, that is making the cell grow spontaneously and not die. So development of the tumor will require additional factors, as I said before, like maybe genetic alterations or the host immunity. Maybe the person is immunosuppressed or has an underlying condition that is making them immunosuppressed, and hence when maybe infected by a certain virus that causes tumor, then that can also help mediate the process. So neoplastic changes happens at the molecular level and also at the cellular level. So when we go back to our basic understanding of cell division and cell growth at the molecular level, I want us to jog behind and maybe try to remember the different stages so that it becomes a little bit easier for us to understand how some of these viruses mediate neoplastic changes. For example, when a normal cell is just at rest and then it needs to divide, there are some growth factors that need to be present. Then there are different changes that need to take place. For example, there are different stages, like this G1 stage, the growth phase. Whatever happens at that stage is different from what happens in the S phase, also different from what happens at the G phase and also the mitotic phase. And also these different stages, remember, they do have checkpoints. Before a cell moves from, let's say, G1 to the S phase, there are checkpoints to indicate that the cell is fit to move into this specific phase. Similarly to before a cell maybe moves from the G2 phase to the mitotic phase, there are checkpoints to make sure that this cell is fit to move into these different stages. And if these checkpoints are not put into place, or there is something hindering these checkpoints, then that results to a change that causes tumors or the neoplastic changes start happening. So that is at the molecular level, and we will see in the next slide some of what really happens. And then at the cellular level, you'll also see on the next slides what basically happens, how the cell morphology changes maybe, or how the metabolism of the cell changes. So cellular oncogenes are present in all normal cells, and they code for proteins that are involved in regulating cell growth and differentiation. Now we can relate to what I have just said before. Before a cell, grows and divides, it needs some proteins to take it to the different stages. And so examples of some of these oncogenes are the MYC gene, or if you want to call it the MYC gene. And then there also needs to be checkpoints, whereby if maybe the cell has a damaged DNA, then it doesn't proceed, and the DNA probably needs to be either repaired, or if the cell is completely damaged, then it can undergo apoptosis, or it can undergo cell arrest. So there are those checkpoints where now the tumor suppressor gene comes in, and these are used for inhibiting the malignant transformation of these cells. So examples of tumor suppressor gene, we have the RB gene, that is the retinoblastoma gene, and the P53 gene. So now onto the cellular transformation. We've been looking at the molecular transformation at the molecular level, but now at the cellular level, once molecular transformation has taken place now, what happens to the cell? So the morphology of the cells get altered, the metabolism of the cells are altered, then the cells lose contact inhibition in cultured cells. Normally, when you culture cells, they grow in a monolayer singly. So they exhibit contact inhibition. They don't touch each other. But this is different now for malignant cells or for cancerous cells. They form microtumors, and they grow, they lose that contact inhibition content. And if the causing factor is, let's say, a virus that has infected that cell, that is making it transform like that, then now this particular cell starts expressing the viral specific antigen, and it also ends up losing its cell specific antigens that are usually expressed on the cell surface. So now the cells become immortal, so they don't die, they don't go through senescence, they continuously divide, and hence tumors form. So this is what we've just been talking about on introduction. We've been talking about how the percentage of viral infections across human cancers, and we gave some examples of some cancers that are implicated with viruses, and the slide shows a list of a couple of some, like Burkitt's lymphoma, hepatocellular carcinoma, Kaposi sarcoma, and we need not to forget that the virus is not the only factor. There are other factors that also have to be in place. So what are these oncoviruses? They are basically just viruses that cause cancer. And some of the non-oncoviruses are hepatitis C, hepatitis B, the human T-cell lymphotrophic virus 1, human papilloma virus, human herpes virus 8, and the Merkel cell polyoma virus, and EBV, Epstein-Barr virus. So these are some of the non-oncoviruses that cause cancer. So these viruses are categorized in either DNA viruses or RNA viruses. So for the DNA viruses, we have the Parvoviride, whereby we have the human papilloma virus and the Merkel cell polyoma virus. While for herpes viridae, there's the herpes simplex virus 2 and the Epstein-Barr virus, together with the human herpes virus 8. Then for Hepadna viridae, there is hepatitis B virus. Going on to now the RNA viruses, we have the Retroviridae family and the Flaviviridae family, whereby under Retroviridae we have HTLV1, and Flaviviridae we have HCV, that is the hepatitis C virus. So these viruses, for example, if we take the DNA viruses, what is their mechanism of action in causing cancers? So for DNA viruses, a portion of the virus gets integrated into the host cell chromosome. We all go back and understand how replication of the viruses happens in the cells. So for them, what basically happens is the portion of their virus gene can get integrated into the host cell. And this, once another gene gets integrated into the host cell, there's potential of, genetic instability, or if it gets integrated into a gene that was functional, then that gene can cause the gene to mutate. And then these viruses can also encode proteins that are usually called oncoproteins that activate proto-oncogenes. So proto-oncogenes are just the oncogenes that we've been talking about, the genes that are normal in cell, and they're usually expressed during cell growth and cell division. Or these oncoproteins can inactivate the anti-oncogenes. So anti-oncogenes, we've also talked about them. They're the tumor suppressor genes. So there are two factors playing in here, whereby these proteins, which we are calling oncoproteins, that are encoded by the virus, can either activate the proto-oncogenes, so which we've said are the genes that are activating the cell growth and cell division. So then if they activate the proto-oncogenes, means there is potentially increased cell division, cell proliferation. And they can also inactivate anti-oncogenes, meaning they can inactivate the tumor suppressor gene. And we saw the importance of tumor suppressor gene. They're involved in DNA repair. And if the cell is so much damaged, it can also kill the cell. So it's just regulating the cell division process. So if these tumor suppressor genes are inactivated by the oncoproteins, then there is also a potential of the cell continuously dividing and continuously growing, and hence the neoplastic process begins. So all these, as we have said, result to genetic instability, and ultimately there is loss of cell growth regulation. So what about the RNA viruses? How do they cause oncogenesis? So we've seen retroviruses use two approaches. So there is the direct action or indirect action. Under direct action for retroviruses, the tumor virus usually introduces a new transforming gene, which we call the V-onc, into the cell. So remember, if you've gone through retroviruses, during their replication, there is a stage whereby they integrate their gene into their host cell genome. So if this virus integrates its gene into the host cell genome and it has encoded for a certain viral oncogene, then that is how it introduces now this new transforming gene. Or they can encode oncoproteins, which are almost identical to cellular proteins. So we've been talking about the cellular growth, the cellular proteins that are there to regulate, cell growth and cell division, or they essentially activate cell growth and cell division. So if this particular virus encodes for oncoproteins that are identical to those of the cell, then this virus, as it keeps on replicating in the cell, it now overexpresses these oncoproteins. So now the cell keeps on dividing and and growing, and that is also a potential of a neoplastic process taking place. Then for indirect action, the tumor virus will alter the expression of a pre-existing cellular gene. So if, for example, the cellular gene was a gene that is encoding for, let's say, a tumor suppressor gene, and this particular tumor virus alters its expression, meaning it's going to inhibit the processes that are essential for a tumor suppressor gene, and hence the neoplastic process again begins. So we'll now go through some of these viruses one by one and try to understand some of the mechanisms that have been described. Some of them are not fully, because some are still under research. Some processes are still yet unknown. So the evidence that has been displayed is what we will try and understand. And we will start with the viruses that cause hepatocellular carcinoma, that is the hepatitis C virus and hepatitis B virus. So what is the mechanism for hepatitis C, for example? So for HCV, the main mechanism of causing hepatocellular carcinoma is persistent viral infection. That is, with persistent infection with HCV, there results to necrosis of the hepatocytes, inflammation, and maybe also cirrhosis of the liver, and finally hepatocellular carcinoma. So most of these viruses that cause hepatitis, people go to chronic infection. I mean, so they will be having an active infection, but a bigger percentage will potentially develop chronic infection. So with chronic infection now, there is that persistent viral infection, because you're literally just infected by the virus chronically. And that is what now damages the liver, because every time then the liver undergoes necrosis and fibrosis, and finally there's potentially formation of hepatocellular carcinoma. Then again, for hepatitis C, there is the hepatitis C core protein that usually interferes with the p53, which we saw earlier as an example of the tumor suppressor gene. We saw there was the retinoblastoma gene, and there is p53 gene. So for HCV core protein, it interferes with this p53 gene. So once you interfere with tumor suppressor gene, then your cell growth and cell division is no longer regulated, and so the cells can just grow uncontrolled. So as we all know, HCV can infect anyone, and some of the high-risk groups include people who inject drugs, the IV drug users, and transplants and transfusions. But this is nowadays is a little bit limited, because before transfusion, the blood has to be screened for the transmissible infections. So this is on the lower side, and also for people with who engage in high risk sexual behavior, together with body piercings and tattoos, and those babies who are born to Hep C positive mothers. So some of the treatment options for hepatitis C, I'm sure you may have gone through some of this, include interferon and ribavirin, but some of these drugs you'll probably learn them more when you'll be learning about, I don't think you've already taken the antiviral class, but when you'll take it, you'll learn the mode of action of some of these antiviral drugs. But just to mention, there's ribavirin, interferons that are used for treatment of hepatitis C infection, and currently there are antivirals that are called the direct acting antivirals that are used to treat HCV infection, and the treatments are usually genotype specific. For someone who has already gotten hepatocellular carcinoma, they have to undergo either liver resection or transplantation, or go undergo chemotherapy. So hepatitis B also causes hepatocellular carcinoma, and the same main mechanism of action that I've been talking about, persistent infection, when now someone is chronically infected and the liver undergoes necrosis, constant inflammation, fibrosis, and finally, they end up developing hepatocellular carcinoma. So the oncogenic potential for Hep B, hepatitis B virus, it codes for the HBX gene. And this HBX gene activates a variety of transcription factors and signal transduction pathways that are involved in cell growth and cell division. So when someone is chronically infected with hepatitis B, the constant presence of this gene, and then there is constant, there's constant activation of these transcription pathways that will lead to constant activation of the cells to, divide and grow, and the neoplastic process begins. So again, who can get hepatitis B? Anyone. And the high risk, behaviors like high risk sexual practices, getting into contact with body fluids, infected blood, whether wet or dried, person to person in early childhood, especially through body piercings and tattoos. So those are some of the transmission dynamics for hepatitis B. And treatment, it was the first major human cancer that was can be treated by vaccine. So treatment is also genotype specific, and it involves antivirals and interferons. For people who've already developed cancer, liver resection or transplantation is done. So now we move into this RNA virus, the human T-cell lymphotrophic virus 1. And which cancer does it cause? It causes adult T-cell leukemia or lymphoma. And most patients usually die within a year of diagnosis. So this cancer is very rare. And the latency for this cancer is about up to 40 years. So, and also the number of people that get infected by this virus that is finally get the cancer is also a very small percentage, only up to around 3%. So it shows that, its mechanism for carcinogenesis is a little bit inefficient. So maybe let's just go through its carcinogenic potential and say, but it's a very rare cancer anyway. So this virus encodes for the TAX gene and HBZ gene. So these two genes have the potential of they have the potential of increasing the transcription for the genes that are encoded for T-cell proliferation. So the transcription factors that have their genes encoded for T-cell proliferation are activated by this TAX gene and the HBZ gene. And also these two genes have the potential to interfere with the DNA repair pathways. So those are the two basic ways of this virus having the ability to cause adult T-cell leukemia and lymphoma. So far we've seen that the major mechanisms that this virus is used are either one, to increase or activate the oncogenes, or to inhibit the tumor suppressor gene. So it just depends on which mechanism, which particular virus is using. So again, treatment for this virus will involve the reverse transcriptase inhibitors, for example, tenofovir, because it's a retrovirus. So I hope we've gone through the Retroviridae family and learned about its replication. Then you'll be able to understand how the drugs act. So for treatment of cancer, again, there is chemotherapy and the antivirals like zidovudine, and some immunotherapy or bone transplantation, because the virus, it's a leukemia, so it infects the blood cells. So who is at risk? Anyone. And high risk in African American groups. It's endemic in Japan, South America, and Asia, and it's usually transmitted via blood. So another DNA virus now, the human papilloma virus, which causes cervical cancer and is also associated with oropharyngeal cancers and anal and genital cancers. Again, who can get HPV? Anyone. And it's the second most common cancer in women. It can take up to 15 to 20 years to develop. And the risk factors include multiple sexual partners, tobacco use, and immune suppression. So HPV have about 70 genetically distinct genotypes. So for example, we have HPV 1, 2, 4, and 7 that are implicated for causing benign squamous papillomas, that is the warts. And then we also have the high risk HPV 16 and 18 genotypes that are implicated for squamous cell carcinoma of the cervix, the anogenital region, and then the head and neck region. Then there is the low risk, which includes HPV 6 and 11. And these ones are implicated in genital warts of low malignant potential. So we might ask ourselves, like, why the variation in the cancers? Some are not even cancers, some are just benign, and others there's low risk, and others is high risk. So for benign warts, the HPV genome is not integrated. So it remains in the episomal form. So it doesn't get integrated in the host genome. While for the cancers, like for the squamous cell carcinoma of the cervix and the genital warts, the HPV is integrated in the host genome. So then viral integration is usually random, but integration is clonal. It gets integrated into the specific clone of cells. So this integration of the HPV genome usually interrupts the viral DNA within the E1, E2 open reading frame, and hence there is loss of the E2 viral suppressor. E2 viral suppressor is responsible for, it attaches to the promoter of these specific viral proteins here, down here. There is E6 and E7. These are viral proteins that are essential for generating the neoplastic process. So this E2, when it attaches to the promoter of these two genes, then it represses them. So then the E6 and E7 genes of HPV will not be expressed. But in a case where E2 is repressed, so it cannot attach to the promoter site of E6 and E7, then E6 and E7 are repressed and the proteins are produced, and now they can initiate the neoplastic process that causes the cancers. I hope that is clear. So now these oncogenic proteins that I was talking about, the E6 and E7, they have the potential to inactivate the tumor suppressor gene, They can activate the cyclins. So cyclins are proteins that activate they are pro cell growth and pro cell division. So they increase cell growth and cell division. And then these oncogenic proteins, that is the E6 and E7, will inhibit apoptosis and cellular senescence. So they are ideally, inactivating the tumor suppressor gene, but activating those processes that increase the cell division and cell growth. So the result is immortalization of cells that continuously divide and don't die. Then there is increased cell proliferation and genomic instability because of the integration. Good news is there is vaccine available for HPV. So for ladies like us who haven't gotten to, some of us have gotten to 26 years and above, but for those people who haven't been to 26 years, it's advisable to get the vaccine for HPV. It will protect you against getting the cancer. So we also have the HHV8 that causes sarcoma. And this virus, how do Kaposi sarcoma appear? They look red and purple depending on the skin tone of the person. For the lighter versions, they look reddish, and the darker skin tones, it looks purple, like purple growths on the surface of the skin. It can infect anyone, but for those people who are immunosuppressed, or people who are going through chemos and whatnot, then they can potentially, be infected by this virus and get Kaposi sarcoma. So that explains why HIV patients and transplant patients or elderly and chemo patients can easily be infected with this virus. So because normally the virus is so much associated with AIDS patients, infection is widespread in sub-Saharan Africa, because as we all know, sub-Saharan Africa bears like 70% of the burden of HIV globally. So it's sexually transmitted, it infects the lymphocytes and establishes latency. It inhibits the p53 tumor suppressor protein, so that is its oncogenic potential. And cell lysis allows it to escape and infect surrounding cells. What are some of the prevention strategies? Safe sexual practices. For treatment, when someone has already developed cancer, there's surgery, radiation, and chemo. But if someone was infected because they already had maybe HIV, then it's noble to first, treat the AIDS-related cause first. So Epstein-Barr virus, which causes Burkitt's lymphoma. Sorry, I just got distracted a little bit, but we can proceed. So Epstein-Barr virus causes Burkitt's lymphoma or nasopharyngeal carcinoma. And in HIV patients, the infection is usually associated with CNS lymphomas. And 90 to 95% of people get infected in childhood with no symptoms. And EBV causes infectious mononucleosis in adolescents, which is a self-limiting, symptoms. At this stage, it barely causes the cancer. But once it becomes latent in the B cells, it has the potential of causing cancer, and its carcinogenic potential is it encodes for the latent membrane, the LMP, especially LMP1, and EBV nuclear antigen, which is also encoded by this particular virus. So this latent membrane protein acts as CD40 on, so back to basic, I don't know if this is basic immunology, but when B cells express a CD40 on their surface, T cells will send signals that will cause proliferation of the B cells. So this LMP acts like the CD40. So then the B cells will start proliferating and growing even without the signal of the T cell. While for how the EBV antigen, nuclear antigen acts, is that it can activate some CDKs. CDKs and cyclins are for cell growth. So EBV nuclear antigen can activate CDKs and hence activate cell growth and cell division. So that is how the carcinogenic potential of Epstein-Barr virus takes place. So finally, we have the Merkel cell polyoma virus, which causes the Merkel cell carcinoma. And for this virus, the exact mechanism is not known yet. And there is no vaccine or treatment for the virus. But once the person gets Merkel cell carcinoma, they can get surgery or radiation.
Dr. Barclay, we can't hear you. It seems you've frozen.
I'm sorry, class. My network is terrible. So this was the last slide that I was talking about, but we'll go through. So unfortunately, I can just see on my charts that your questions were kind of nullified because I've just joined and now I'm not able to see the questions. So if you don't mind, you can type them back and then we'll tackle them once I finish this slide. So, so the Merkel cell polyoma virus causes Merkel cell carcinoma, and the exact mechanism of the formation is unknown, but there is no vaccine for the treatment of this virus also. And for the carcinoma, surgery and radiation is recommended, and chemo for, palliative care and shrinkage of the tumor. So that's all for the Merkel cell polyoma virus. It's still a new virus that a new mechanism that is under study on how it causes, carcinoma. So I had put this question for us just to see if at least we came out with something about oncogenic viruses. So it's just basically tells us to list how many, to list human oncogenic viruses and their associated malignancies, and list few vaccine preventable viral tumors that we know. And maybe later you can read more and write short notes on some of the mechanisms that this virus is used to generate oncogenesis. So that is all. If you would want to maybe write answers for the first and the second one on the chat, we can go through them, but I'd advise you read more on the last bullet. So I'll go ahead and check on the questions. So someone says LMPs act like CD4. Antigens acting B cell proliferation without any T cell activation. I think this person is just trying to do further explanation, which is true. Victoria says no, Burkitt's lymphoma is a B cell lymphoma presenting as a tumor of the jaw, while nasopharyngeal carcinoma is cancer of the epithelial cells, which is true, because I said the topic was EBV and how EBV causes viruses. So I mean cancer. So the list of cancers that was written there was Burkitt's lymphoma and nasopharyngeal carcinoma. Burkitt's lymphoma is a lymphoma for B cells, while nasopharyngeal is a carcinoma for epithelial cells, which is true. So the LMPs act as CD40, which on normal occasions cause T cells to proliferate B cells. Yes. Is CMV also oncogenic, as it can also cause mononucleosis? No. Remember we said if the virus infects you, doesn't mean that you're going to get the cancer. So mononucleosis is self-limiting. So if you get EBV or you're infected by CMV, if your immune system is intact and you will clear the virus, so you'll just get the minimum symptoms of which is what we call the mononucleosis, the fever, the rash, whatnot. That is what you will get, but doesn't mean that you will develop cancer. So when it now becomes latent on the B cells, so some of those latently infected cells are the ones that now can be transformed to be carcinogenic, but CMV doesn't cause Burkitt's lymphoma. So hence, the uncontrolled B cell proliferation will cause Burkitt's cell lymphoma. True. I think T cell proliferation, not B cell. So maybe just to explain it further. So Epstein-Barr virus codes for LMP1, yeah? So LMP1, let's say, mimics CD40. So when B cells express CD40, the T cells usually send signals for proliferation of the B cells. All right. So now, in this case, this B cell that has been infected by EBV is not expressing CD40, but it's expressing LMP all the time, because the virus keeps on replicating on the cell. So it keeps on expressing the LMP, which is acting like CD40, and hence the B cell keeps on proliferating. Hence it's gives a potential of causing cancer, which is the Burkitt's lymphoma, even without the T cell sending signals to the B cell. So I hope that makes sense. All right, so LMPs act like CD40 co-stimulatory molecules produced by T helper cells that stimulate B cell proliferation without the need of T helper cell activation, hence uncontrollable proliferation of B cells. True. So we already said mononucleosis is not cancer. Clarify on action of LMP. I've just clarified. I hope it makes sense. If it doesn't, please just write again that it doesn't make sense. Then I will do further explanation. Burkitt's lymphoma is a B cell lymphoma presenting as a tumor of the jaw, while nasopharyngeal cancer is a cancer of the epithelial cells. True. Infectious mononucleosis I don't know what that means, to lymphocytosis, which is featured with T cell proliferation. I think it's just the explanation of the same thing we've been talking about. How does the eBNA work in causing cancer? So as I said before, let's go back to to cell division and cell proliferation. Before cell division and cell proliferation takes place, they are the proteins that, are like growth factors that make the cell start proliferating. So these proteins include cyclins and CDKs. So these CDKs, when they are continuously activated, then the cell continuously grows and continuously proliferates. So the function of EBNA here is to activate, so it activates the CDKs. So once the CDKs are activated, then they activate the whole process of cell growth and cell division, and hence the neoplastic process begins. All right. So I think there has been a differentiation of Burkitt's lymphoma and nasopharyngeal carcinoma there, and it's about understanding what is a lymphoma and what is a carcinoma. There was a very good explanation of nasopharyngeal carcinoma. It's a carcinoma of the epithelial cells of the nasopharynx, and Burkitt's lymphoma is a cancer of the B cells. It infects EBV infects B cell, hence potentially there's a formation of that cancer. So cleared by the immune system at Calvin. I don't understand that. Is breast cancer caused by a virus? As per now, I will say no, but if you have probably an article or something that proves that, you can share with us, because we can say no today, but a few years or few times from now, someone comes up with something, evidence that shows that there's a virus that causes this and this. But as per now, I haven't come across any virus with evidence of causing breast cancer. So does something to CDKs which cause cell growth, maybe it's unusual growth, and some I don't really understand what someone was trying to write. So if you feel that I haven't answered the question, just put it again on the chat, so that in a way that I can that is understandable, so that we address it. What about HIV? Isn't it oncogenic? So for HIV, there is no sufficient evidence that says that HIV is oncogenic. But as we have seen, when someone is infected with HIV, then they become immunosuppressed, and some viruses that take advantage of that and cause cancers, other cancers, but it will not be HIV directly causing the cancer. And an example of that is the human herpes virus 8 that causes the Kaposi sarcoma. Burkitt's lymphoma, translocation, 70%, 75% of cases, reciprocation, translocation involving CMYC gene, chromosome 8, and heavy chain, IG locus, chromosome of X. There's a lot of, is this biochemistry or what here? But essentially, I said there is no single mechanism of, the cancer forming. There may be several factors, and so long as there's evidence that these specific factors causes this and this and this, then there's no problem. I may have not written all the oncogenic potential for each virus, but you can go and read along the same lines and get more information. But just to talk about the CMYC gene, remember I mentioned the CMYC gene or the MYC gene as one of the oncogene. And we say that the oncogenes are once the expression takes place of the oncogene, the proteins are expressed, and these proteins are the ones that normally cause cell growth and cell division, like they are pro-cell growth and pro-cell division. So once these genes are not regulated, they keep on being activated, because as much as they need to be activated for cell growth and cell division, there needs to be regulation. So if they're not regulated, then there is the potential of the cancer forming. What about Hodgkin lymphoma caused by, what causes it? Maybe you can share with us which, because Hodgkin lymphoma is a wide group. It's not like specifically, let's say, Burkitt's lymphoma. It's a wide group of lymphomas, and there are smaller, lymphomas underneath. Hodgkin is just a bigger name. So maybe you pick a specific type of Hodgkin lymphoma and explain to us how it causes it, Roy. We would be glad to learn on the same. All right, kindly repeat on the explanation on how weakly oncogenic, that is indirect transformation, RNA tumor virus causes cancer. We'll go back to that specific slide and do that explanation. Before, let me just go through the charts, and then that is the last thing we will do. So just hold your thought there, Grace. So you talked about HSV, which type of cancer does it cause? So for HSV, it has been shown, though it's epidemiologically shown, that it's associated with cervical cancer and also prostate cancer. But now, sometimes these are just statistics, and they haven't really shown the direct association. You see, like how if we are talking about a specific virus, we know that it encodes for this specific gene that does this and this and that. So that has not been shown on HCV, but it has been shown to be associated with, prostate cancer and cervical cancer, but not that it doesn't cause them. Like that direct causation hasn't been demonstrated yet. Okay. So I've just talked about HSV. We can read more articles on the same. So for Hodgkin lymphoma, I said Hodgkin lymphoma is there are so many types of lymphomas under Hodgkin, so we need to know which particular one someone is talking about. So we talked about the difference between Burkitt's lymphoma and nasopharyngeal carcinoma. So are viremic oncogenic viruses likely to metastasize if treatment is delayed and dissemination occurs through the body, or they will only increase the carcinogenesis locally to the specific sites of replication? So I think depending on what type of virus it is, if it's a type of, what type of cancer it is, if it is a cancer that will metastasize, then it can, if it's a cancer that is just gets localized to a certain region, then it will remain localized. So I don't think there is a direct relationship with once the virus causes the cancer. Now, whether the virus will determine if that cancer will metastasize or not. What do you mean by episomal form? The benign ones for HPV. So when you say episomal, we mean the genome will just replicate maybe in the cytoplasm, like how the plasmids do. If you understand how the plasmids work, they are genetic materials that are not in they are mostly in bacteria. They are not in the nuclear, they're just in the cytoplasm, and they can replicate independently. So in this case also, this genome of the virus will just replicate without it integrating itself into the host genome. So that's the difference why we'll say it's episomal. It will replicate, but will not integrate itself into the host genome. So someone says, I don't really think viremia plays a significant role in due to their selective tropism, both in infection and transforming ability. Is Burkitt's lymphoma global, or some literature refer to it as African Burkitt's lymphoma? Does it mean it's an African thing? It's mostly endemic in Africa, and some Asian countries. So some of these things, it goes back to other factors, other epidemiological factors also come into the interplay, because some things happen in Africa, not because it's an African thing, but because maybe our infrastructure and our health facilities and all those other interplays are not in place. And so that's why we experience such kind of burden as compared to the West. So some conditions remain endemic in Africa, yet they can also infect other people, but in other places they are controlled, if that makes sense. So Mohammed says disease results from an injury to the DNA of a lymphocyte type of white blood cell. The damage to the DNA is acquired, occurs after birth rather than inherited. The altered DNA in the lymphocyte produces a cancerous change that, if untreated, results in uncontrolled growth of cancerous lymphocytes. The accumulation of cancerous lymphocytes results in tumor masses that are found in the lymph nodes and other sites of the body. So it's talking about Hodgkin's lymphomas. So as you see, his explanation is very general. So when you say Hodgkin's lymphoma, you're talking about, it's so general in that you're saying it's a cancer that involves the lymphocytes or the white blood cells or something like that. So all these other cancers that will involve the lymphocytes will fall under that, and it can either be Hodgkin's or non-Hodgkin's. So there is also a difference. They all infect lymphocytes, but there's a difference between the Hodgkin's and non-Hodgkin's. So I'll let you go and find the difference between the two in terms of how they appear, the cancers appear, or how, and also in terms of diagnosis. When they are diagnosed, how does someone know that this is a Hodgkin's lymphoma and this is a non-Hodgkin's lymphoma, though they're all cancers that are affecting the lymphocytes. So how does alcohol use fastens hepatocellular carcinoma for HCV? So alcohol use. So alcohol, when I'll just answer this from my thinking. When you take in alcohol, and alcohol, there are toxins from the alcoholic content, then the toxins usually they are, how do we say it? They are all filtered in the liver, right? Something like that. So you make your hepatocytes susceptible. You've already put the hepatocytes at a place whereby once they get infected by something else like an HCV, they're already at risk, because they have been working so hard to intoxicate you. And if I may use layman's language, they are kind of exhausted or something like that. They're not like hepatocytes for someone who does not take in a lot of, alcohol or a lot of intoxicated stuff. So if I can explain it from that point, and also other factors interplaying, then that puts you at a higher risk. Same with smoking, because you're taking in, contents that will overwork your liver, if that makes sense. So I think let's go back to the very first slide. It wasn't the very first, it was some slide on top here about RNA viruses that Grace wanted an explanation on. Just a second, we see which one it was. I think it's this one. So we'll just explain the whole slide again, so for us to get a glimpse of how the retroviruses cause oncogenesis. So, and one of the retroviruses that we talked about that cause oncogenesis is the that causes ATLL is HTLV1. So through direct action or indirect action. So for direct action, the tumor virus will introduce a new transforming gene, that is the V-onc gene, into the cell. So I tried to explain this by saying we remember the replication. If we can relate it to the replication of HIV, whereby there is a step for the virus integrating its genome to the cell. So let's use that as an example, because this is also a retrovirus. So it integrates itself into the host cell genome, and if the genome of the virus had an oncogene, then it transforms the cell now. I hope that makes sense. Or the virus can encode oncoproteins, which are almost identical to the cellular proteins. So some of the cellular proteins that are involved in cell growth and cell division, if the virus encodes an oncoprotein that is identical to those or works like those ones, then as the virus continuously replicates, then there is overexpression of these products that will now continually stimulate cell growth, because you remember the virus is continuously replicating, lysis of the cell, so many viral virions goes infects other cells. So there are so many of these oncoproteins already in the cell. So there is overexpression. And remember, they are acting like normal cellular proteins that normally stimulate cell growth. So now that overexpression then now will cause increased cell growth and cell division. Then for indirect action, now the tumor virus alters the expression. Sorry. Where did the slide go to? Okay. So for the indirect action now, the tumor virus will alter the expression of the pre-existing gene, because let's say, for example, in HTLV1, when the virus is integrated into the cell, then where it is getting integrated, there was another gene there. And this gene was potentially maybe a tumor suppressor gene, and it has gotten integrated there. So means it will alter that pre-existing gene, and this gene will potentially not be expressed. And if its function was to suppress the tumors, it's not going to do that. So in that case, that is also a form that it's now going to initiate the neoplastic process. So I hope that makes sense. and giving an example of also HCV core protein, whereby we said the HCV core protein will interfere with the P53 tumor suppressor gene, if you can remember. So let's go back to, you talked about RB and P53. My question is, what WTI and DC are insignificant in tumor suppression? What is WTI and DCC? Maybe write it down there so that I know. Then, yeah, like the guy who the virus is named after first did his first studies about the virus in Ugandan children, the cancer. So I think you're talking about Burkitt's lymphoma. So WTI, please write WTI in full so that I know what you're talking about. If alcohol overworks the liver, does it mean it acts as an exercise to the liver? And this person calls him or herself Bunyaviride. What a nice name. Roy B lymphocyte lineage cells with crippled immunogene gives rise to Reed-Steinberg cells, which are malignant Hodgkin's. So there's someone trying to give a difference between non-Hodgkin's and Hodgkin's lymphoma there. All right, Wilms tumor something. Saw it on Robbins pathology for pediatric cancers. Is it involved with viruses? Because we have so many cancers and not all of them are directly associated with viruses. That's why we had a list of those, and we said about 20% of the cancers, so not all cancers. But if it is, maybe you can help educate us. More knowledge is good for us. Okay. Okay, so this Wilms tumor gene, Kamunya will probably tell us how it's associated with which virus and what's the mechanism of it causing a cancer. For HHV8 is the second slide, also in relation to Kaposi sarcoma. All right, let's go back and see your question. Should be somewhere here. So this one, the second slide after, because that's Epstein-Barr now. This slide is still talking about Kaposi sarcoma, so it's still the same. If that was your question, it's just giving details of how it's transmitted, potentially how it causes cancer, and some treatment mechanisms. So I hope that answers your question. Oh, the one written Epstein-Barr virus, no. So that is now on Epstein-Barr virus. There is no association with HHV8 now. So the next one is Epstein-Barr virus. There's no association. So do we still have more questions? Oh, which textbook do you use for virology? There was this book I put on the, can you check on the very first very first presentation I made on lab diagnosis. There was a textbook I wrote there. I can't remember the name right now, but we are going to have another class with you. I'm going to have another class with you on Friday. So I'll get the name of the book, but I also use lunch review of medical microbiology. You can check it out. I think you know that one, because all medical students, I think, know lunch review of medical microbiology. I don't know what edition I have right now, but you can check it out. For the other one has, I put it on the references for lab diagnosis slides. Please check them out. So are we satisfied? Did we get something? Or did we learn something today? Please tell me on the slides so that I know. All right. So I'm glad we were able to learn something. I'll have another lecture with you, I think, on Friday. Yeah, Friday, but I can't remember. It's on viral isolation and culture, and I think we tackled some of those on the lab diagnosis class. So we'll probably not really brush through it, because you remember I told us to go and read about shell viral cultures and insect cultures. So please read about those, because we had done, and any modern technology on virus isolation and culture. Read about that. We'll discuss and learn from ourselves. So yes, I'll share the slides with Kelsey, and I think are we good to go now? Can we call it a day?
Yes.
All right. Okay. All right. So see you guys on Friday at 2 p.m. and enjoy your day.